Today, our speaker today is Dr. Matthew Rasband, Matt for us. Matt is a professor at the Baylor College of Medicine, and he's also director of the graduate program at the Baylor College. And the last year, he was the chair of the chair of the school of the joint meeting of the American and International Society for Neurochemistry that was organized in Montreal, in Canada. He said that he was really very passionate for physics, and for that he has a bachelor in physics and then a master and a PhD in biophysics. Fortunately for us, 20 years ago, he landed in the neuroscience field, and from then he has developed a very brilliant career publishing more than 100 papers in the most prestigious journal really in the world. Let me tell you that he's also very passionate in motorcycles, and he has around his motorcycles from Mexico, Spain, Greece, and we hope next year that you rode your motorcycle from Argentina.
I would love to.
I met Matt when he was really a very, very young scientist in a meeting of the American Society for Neurochemistry, and soon he won a very prestigious award from the American Society, the award in honor of Jordi Folsby, and now he has a lot of awards. In 2005, I have the opportunity of organizing a school supported by IBRO, and I organized this school in the province of Buenos Aires in Pinamar, and Matt was one of the teachers. At that time, Matt already worked in the node of Rambier, and remember very clearly that before starting his talk with the student, he said to the student that he loved so much the node of Rambier that at night he used to dream of them. But he don't dream in black and white, but in color. I never forgot this introduction to this class. Now, Matt is working in his lab in understanding how neurons specifically cluster the voltage-gated sodium channel at the site where excitatory signals are integrated and propagated to the node of Rambier. Mauricio, as you know, he was a postdoc at the Matthew's lab, and both of them has done a very good job and published a paper in Cell, very excellent paper. And now, well, I go directly to the seminar on the functional organization of the axon. Thank you, Matt, for accepting our invitation, and we wait for your talk.
Okay, Juana, thank you very much for that kind introduction. Can you hear me fine still, Jorge?
Yeah, perfect.
Yeah.
Okay. So I have very fond memories of visiting Argentina. I've been there twice to Buenos Aires.
Mm-hmm.
And, the first time was for the International Society for Neurochemistry, the ISN international meeting. And then the second time, as you just mentioned, for the IBRO meeting. And I remember how much I enjoyed interacting with all of the students there and how beautiful Pinamar was. I still tell my wife that I want to buy a place in Pinamar someday and retire in Pinamar. I just need to learn Spanish. So I today am going to tell you about the functional organization of axons. And to begin, I'd like to ask you to think about this problem. At this very moment, we are communicating over tremendous distance. And this distance, the way that we do this is I am on a computer that connects to a network. And the way that you see my image is that this network is connected to what is referred to as terminal equipment. And that terminal equipment then connects to a cable. In our modern world, these cables are fiber optic cables, but it used to be that, they were they were hardwired, just wires. And I'll show you examples of those in just a moment. And these cables, they span the globe. They cross oceans. They connect between here and Argentina. And some features of these cables that are very important is that these cables, they must be armored to withstand mechanical forces to which they're exposed. So they have to have a great deal of tensile strength. They have to be lightweight, and they have to efficiently conduct the signal. Although cables can do this quite well, because the distances are so long, there's still noise and interference that is introduced into the system. And so what engineers do is they build what are called repeaters in these cable systems. And these repeaters, they function to condition and regenerate the signal. Our nervous system is built on exactly the same principles. So all of that network input is analogous to the synaptic input, the synaptic input that arrives here in the somatodendritic domain. The terminal equipment is like the axon initial segment, where sodium channels are clustered in high density and integrate that synaptic input. The action potential is initiated and then begins to propagate down the axon, which is analogous to this, this cable system. And even though the axon is able to conduct the action potentials efficiently, there is still loss in the system. And so the signal must be regenerated at repeaters that in the mammalian nervous system are called nodes of Ranvier, where the signal is regenerated. Another important point that I want to emphasize is that just like the transoceanic cable, it must be armored and have great tensile strength. The axon also has to have great tensile strength because it runs along our joints and our ligaments. And as we run or we move, those axons, they experience tremendous mechanical forces, tension and torsion that must be resisted so that the axons themselves don't break. And today what I want to talk to you about is these mechanisms for how the nodes of Ranvier are assembled and maintained. And I'll also talk a little bit about the mechanisms that control the stability of axons and give them their tensile strength. So just briefly, I remind you about the functional organization of axons. They have an axon initial segment. Nodes of Ranvier reside between the internodes made of myelin. And the axon initial segments are intrinsically specified by neurons. That means they do not require any help from another cell type or any extrinsic factor. On the other hand, a node of Ranvier will never form without the help of myelinating glia. So despite the fact that these two structures are very similar in their molecular composition, they are quite different in their mechanisms of assembly. An axon initial segment is built from the inside out, whereas a node of Ranvier is built from the outside in. And so today I'll tell you about, I'm going to focus on nodes of Ranvier, although a lot of my lab, at least half of my lab, also works on axon initial segments. And I'll just mention that some of the lessons that we've learned at nodes also apply at sites of synaptic contact. But again, that may be the subject for another day. So let me just show you what these repeaters look like. This is an actual repeater from a transatlantic cable system. And I liked this one because the colors matched my node of Ranvier here. And you can see this beautiful node with the overlying myelin sheath. Here's the gap in the node, and the green, these are the voltage-gated sodium channels. And this cluster of voltage-gated sodium channels is only about 1 micron in length, despite the fact that the axon itself, in you and me, it can be a meter or more in length. So this represents tremendous control and fidelity for the organization of these membranes. Potassium channels of the KV1 family are shown in red. So how about these cables? Let's look a little bit more closely at these cables. I tried to draw a comparison between the cables and the myelinated axon. you can see that this was what a transatlantic cable looked like back in 1858. This is one of our modern fiber optic cables. But if you look at them, something that you'll notice is that they all have insulation. They all have a conducting core. And they all have armor. You can see these are steel wires that provide the tensile strength to these transatlantic cables. And in exactly the same way, a myelinated axon has insulation, it has a conducting axoplasm, and it has a cytoskeleton comprised mainly of spectrans and anchorins that function to confer this mechanical strength onto myelinated axons. This spectrin cytoskeleton, over the last several years, we've come to appreciate that it has a remarkable organization. And this was revealed using super resolution microscopy. This cytoskeleton consists of spectrin tetramers that are spaced by rings of actin that line the inner surface of the myelin sheath. And these spectrin tetramers, they can function as a kind of spring to flex and allow the axon to withstand the mechanical forces to which it's exposed. So let me give you just a brief primer or introduction to anchorins and spectrans. So all spectrans consists of alpha and beta subunits. 2 alpha subunits combine with 2 beta subunits to form a tetramer. These spectrans, they have many spectrin repeats, and each repeat functions like a spring. There are 3 anti-parallel alpha helices that can stretch to allow the spectrin molecule itself to stretch and flex. There are 5 beta spectrans, beta 1 through beta 5, and the main beta spectrans that we find in the nervous system are beta 1 through beta 4. There are 3 anchorins. Anchorins always function together with spectrans. And what the anchorins do is they link membrane proteins to the underlying cytoskeleton. In vertebrates, in you and me, there are 3 different anchorin genes, ANK1, 2, and 3, and the proteins correspond with anchorin G, B, and R. All 3 are expressed in the brain. So I would like to impress upon you the fact that anchorins and spectrans are very important for disease. There are human mutations in all anchorins and in all spectrans, and they lead to a variety of nervous system diseases, from severe cognitive dysfunction to axon degeneration to spinocerebellar ataxia, and the list goes on and on. Anchorins and spectrans are also, and so these are human mutations, pathogenic variants that arise spontaneously or are inherited. But anchorins and spectrans are also very potent substrates for calpain-mediated proteolysis that can occur as a consequence of injury. For example, this is showing brain membrane, an immunoblot brain membrane homogenate with an injured and a control brain. And you can see this is labeled for alpha-2 spectrin. And this spectrin on the injured side of the brain, with increasing times, a breakdown, a proteolytic fragment forms. And this proteolytic fragment is a consequence of the activation of calcium-dependent protease calpain. In the absence, so what is the consequence of this kind of disruption? Well, the consequence is, as I said, these spectrans are thought to be important for maintaining axon integrity. And in fact, you can see this here. We generated alpha-2 spectrin conditional knockout mice, and just comparing the control to the alpha-2 spectrin knockout. These are dorsal roots, and you can see widespread axon degeneration, much smaller axons, and significant pathology in these mutant mice. So it's not difficult to imagine how an injury that results in proteolysis of this spectrin and anchorin cytoskeleton can cause very severe axon degeneration and neuronal injury. Just as another example, here's an example of several pathogenic variants in a spectrin called beta-4 spectrin. Now, beta-4 spectrin is a particular favorite of mine because it's the spectrin that's found at nodes of Ranvier and axon initial segments. So a few years ago, we identified 5 different families that had a variety of mutations. Some of these were compound heterozygous mutations, some homozygous mutations, and these mutations arose throughout the different, so there are 2 main beta-4 spectrin splice variants, and these mutations arise throughout throughout spectrin. And these pathogenic variants can be truncations, they can be frameshift mutations, a variety of different things. But all of these patients, all of these children who have these mutations, they have several common features in terms of their pathology. All of them have profound intellectual disability, profound weakness, areflexia, and axonopathy. So their motor axons degenerate. Interestingly enough, their sensory axons appear relatively normal, but you can see disrupted nodes of Ranvier in some of these pathogenic variants. And on muscle biopsy, you can also see degenerating myofibers. So these kids are very, very sick. So I just wanted to highlight this to emphasize to you how important these spectrins are for normal nervous system function, especially as they relate to axon initial segments. You can see beta-4 spectrin labels the axon initial segment here, and for nodes of Ranvier. And beta-4 spectrin would be co-localized with sodium channels in this particular image. So for the purposes of today, I'm not going to talk about the axon initial segment, although it's a fascinating subject and has inspired many of our thoughts about how nodes of Ranvier are assembled, but I'm going to confine my comments to nodes. And I want to, at this point, acquaint you with some of the terminology that I will use. The node of Ranvier itself is the site where the sodium channels are clustered. Immediately flanking that is the site where the myelin sheath attaches to the axon, and this is called the paranode. Beneath the myelin sheath, not the internode yet, but a short region about 20 microns long, is a region called the juxtaparanode. And this is characterized by a high density of voltage-gated potassium channels called Kv1.2. And each of these membrane proteins is found in the axon. One of the things that I want to highlight to you is this incredible fidelity, how precise these membrane domains are maintained by the overlying myelinating glial cell. And so I have for now 25 years been fascinated by the molecular mechanisms that underlie this kind of organization. So how do you study only nodes of Ranvier? Since most neurons that we think about have both an axon initial segment and a node of Ranvier, if you disrupt any, and as I said, the molecular composition of these 2 domains is very, very similar. And so if you disrupt one protein, it's going to affect both initial segments and nodes of Ranvier. So how can you study and focus on nodes themselves? Well, fortunately, we have available to us a system or a neuron that has no axon initial segment, and that is the peripheral sensory neuron. Peripheral sensory ganglia are pseudounipolar neurons. They're myelinated with peripheral and central branches, and they have many, many nodes of Ranvier, but they don't have an axon initial segment. So we can use a very nice Cree line called the Advillin Cree, which results in recombination only in sensory neurons. And this allows us to do loss of function studies in a neuron that lacks an axon initial segment. Okay, so many of the experiments that I'm going to show you are using these sensory neuron specific Cree mice to uncouple the role of these proteins at the axon initial segment from their role at nodes of Ranvier. So over the last 20 years, my laboratory and several other laboratories have worked very hard to define all of the molecular interactions that exist at nodes and their associated domains. This slide is sort of our current understanding, state of the art knowledge. And it's not essential that you know all of these things, but what I would like to highlight to you is that if you look in each of these domains, you'll notice that there are spectrins in all of these domains. beta 4, beta 1, beta 2, and beta 2, alpha 2. And so in each of these domains, oh, as well, notice anchorins, the associated proteins that link to the business molecules like the sodium channels or the cell adhesion molecules that mediate the interaction between the myelinating glial cell and the axon. So what I'd like to do today is I want to focus on the functions of these anchorins and spectrans and show you how these gene families have been repurposed multiple times in order to assemble and maintain this critical structure called the node of Ranvier. So where should we start? Let's start at arguably the most important interaction, and that is the interaction that is responsible for the clustering of sodium channels. So when we started these projects, this was our view, or as I'll say, the old view of what we knew about molecular interactions. And we knew, or we thought we knew, that anchorin G was essential for the clustering of voltage-gated sodium channels. So Tammy Ho in the lab, she generated a conditional knockout of anchorin G, and we expected that these mice would have no clustering of sodium channels. But when we looked in the sensory roots of these anchorin G conditional knockouts, you can see that anchorin G is completely gone, but much to our surprise, sodium channels were still highly clustered. This was very surprising to us, and because all of the reviews and the papers that had been published to this point argued that anchorin G was absolutely essential. But these mice, they were completely normal. Sodium channel clustering was normal. If you put them on a rotor rod, they were completely fine. If we measure the conduction velocity of their sensory roots, they were also perfectly fine. So how to explain this difference? Well, when you encounter a knockout mouse that appears to have no phenotype, what do we all do? We say there must be some form of compensation. And then we leave it at that and we run away because it's hard to test compensation. Well, in this case, it's not too hard to test compensation because we know there are only two other possible anchorins. So we thought that would be the easiest thing to check. So as I said, there are three anchorins, anchorin B, anchorin R, and anchorin G. And in control roots, you can see that anchorin G is found at nodes of Ranvier. Anchorin B, it turns out, is found at the paranodal junctions in the myelinating glial cell. As I showed you in that cartoon a moment, anchorin G, something remarkable happens. This protein called anchorin R completely replaces the anchorin G and can completely compensate for the loss of anchorin G. This, in my opinion, is a beautiful example, perhaps one of the most beautiful examples of redundancy and compensation in response to elimination of a single protein. And these clusters of anchorin R, they co-localize with voltage-gated sodium channels. And these mice, they are perfectly normal for years. They do not care that they have substituted anchorin R for anchorin G. So as we were doing these experiments, we noticed, and because of time, I'm not going to show you these data, we noticed that at these nodes of Ranvier, they lacked beta-4 spectrin. Remember, this is the link between the sodium channel and anchorin and the underlying cytoskeleton to maintain the structure. I showed you in the children that lack appropriate beta-4 spectrin that they can be very sick. Well, we realized that perhaps just like anchorin G has its preferential binding partner beta-4 spectrin, it may be that anchorin R has a preferential binding partner. Anchorin R, what I didn't tell you, is it's highly expressed in erythrocytes, in red blood cells. Human mutations in anchorin R cause severe hemolytic anemia due to a fragile erythrocyte red cell membrane, so that when the blood cells go through the vasculature, they break. And anchorin R, its preferential spectrin binding partner is beta-1 spectrin in the erythrocyte. So we wondered, in this situation where we knock out anchorin G, perhaps, and it's replaced by anchorin R, perhaps beta-1 spectrin is also compensating. And indeed, that is exactly what happens. So you can look here in these conditional knockouts where beta-1 spectrin is now found at nodes of Ranvier where there's no beta-4 spectrin. So beta-4 spectrin is found at this node, and there's no beta-1, but at this node where there's no beta-4, you see compensation by beta-1 spectrin. And to more clearly show that, Chung-Shin, a graduate student in the laboratory, he developed conditional knockout mice for beta-4 spectrin. And when he knocked it out, so you can see here at normal nodes of Ranvier, there's lots of beta-4 spectrin at every node. But when you conditionally ablate it from the sensory neurons, it's gone. And amazingly, beta-1 spectrin not found at nodes in the control, but now it's found at every single node of Ranvier in the beta-4 spectrin knockout. Perfect, beautiful compensation between beta-1 and beta-4 spectrin. So that suggests that there are these two redundant pathways where anchorin G, when it's lost, anchorin R can compensate, or when beta-4 spectrin is lost, beta-1 spectrin can compensate. And it goes beyond that. It's not just that a single one of these molecules compensates, but both of them compensate for the loss of a single one. So that's a remarkable kind of redundant mechanism to ensure assembly of nodes of Ranvier. Now, you'll see here I have these dotted lines that connect them. I don't have time to show you the data, but we looked at how there are different affinities between these anchorins, sodium channels. They can't just mix and match. Why is it that anchorin G is found normally at nodes and not anchorin R? Well, the reason is because that the affinities, and I've tried to illustrate a stronger affinity with the solid arrow compared to the dotted line, the affinities for these molecules are much higher than these ones. So what do these molecules, so are anchorins required and are spectrins required for normal assembly of nodes? To test this, we constructed double conditional knockouts. We had to remove both anchorin G and anchorin R. So here's the anchorin G loss alone. Sodium channels are still clustered. There's no anchorin G. But when we remove both anchorin G, ANK3 is the gene for anchorin G, ANK1 is the gene for anchorin R. This is a spontaneous mutant is a full anchorin R knockout. And these guys die within about 2 weeks of age due to anemia. But that's enough time for us to see formation of nodes. And you can see the formation of the paranodal junctions, but there's no anchorin G, there's no anchorin R, and there's no clustering of voltage-gated sodium channels. And you can see that more clearly here. So here's the anchorin G single knockout. Sodium channels are still clustered because of anchorin R. But in the double knockout, sodium channels fail to cluster because you must have anchorins in order to cluster sodium channels in myelinated axons. What about the spectrins? So Chung-Shin also generated a beta-1 spectrin conditional knockout. And so we generated beta-1, beta-4 double conditional knockouts. So in the absence of both beta-1 and beta-4, these double conditional knockouts have severe ataxia. They drag their hind legs. They cannot remain on a rotor rod. And when you look at nodes of Ranvier, interestingly enough, during early development, sodium channels are still clustered, so nodes form properly, but over time, the sodium channel clusters are lost. So what this means is that the spectrins are required to maintain the sodium channel complex so here's the quantification. You can see early, they're intact, and then with increasing age, the number of nodes with sodium channels decreases dramatically. So what this means is that these nodal spectrins are required to maintain sodium channel clustering as opposed to the initial assembly. Whereas anchorins, so nodal anchorins are required to assemble the nodal sodium channel complex, whereas the nodal spectrins are required to maintain the nodal sodium channel protein complex. Okay, so I've told you about these anchorins and spectrins at nodes. But there's another molecule here so I'm going to take a little bit of a diversion here and highlight another really interesting protein. Because this is a glial group, I wanted to highlight this interaction here between glial-derived factors in the peripheral nervous system and the central nervous system. All of these glial factors feed into a cell adhesion molecule called neurofacin 186. And this molecule, neurofacin 186, is the binding partner or the membrane attachment site for Anchorance. Maybe you can see it more clearly here. So all of these extracellular matrix, glial-derived extracellular matrix molecules interact with neurofacin 186. It functions as an attachment site for Anchorin G and Spectrans, which then cluster the voltage-gated sodium channels. So one would predict, based on this model here, that neurofacins would be essential to the proper formation of a node of Ranvier and the proper recruitment of Anchorance. So to test that, we constructed neurofacin conditional knockout mice. And amazingly, when we did these experiments, so this is now a Thigh-1 Cree, so these are actually spinal cord axons. You can see that they lack neurofacin, so green is the neurofacin is in green, but sodium channels are still clustered, and Anchorin G is still clustered, and beta-4 spectrin is still clustered. This was a huge surprise. So how can it possibly be that sodium channels can still be clustered in the absence, and Anchorin G still clustered in the absence of neurofacin? Well, actually, the answer came to us as we looked at, right here. The answer came to us through inspiration from some work that Mauricio did. So I want to highlight Mauricio Galeano's work. This was a paper he published when he was a postdoc with me in my lab. And what he discovered is that spectrans and anchorins form a distal axonal barrier that can control the clustering of Anchorin G at axon initial segments. This was a critical insight for us because it turns out, as you can see here, that there are spectrans at perinodes as well. And we thought that perhaps the perinodal junctions, which still remain intact in these neurofacin 186 knockouts, perhaps they function as intraaxonal boundaries or barriers to restrict Anchorin G and beta-4 spectrin to the nodes of Ranvier and thereby cluster sodium channels. So how would we test that model? Well, we were at the same time studying the role of beta-2 spectrin. I've already told you about beta-4 and beta-1, but beta-2 spectrin, we had discovered it at perinodes a few years previously. And so Zhang in the lab constructed a beta-2 spectrin conditional knockout, again, deletion only in sensory neurons. And you can see that these mice have a hind limb clasping phenotype. But the most important thing that I want to draw your attention to is when you look at nodal structure in these beta-2 spectrin conditional knockout mice, they have intact perinodal junctions. So by electron microscopy, one of the cardinal features of a perinodal junction are these electron dense bands. I hope you can see them here. They're these darker structures that are regularly spaced. And those are this complex right here. That's what is this complex, seen at the electron microscope level. And so in these conditional knockouts, the interaction itself was completely intact between the myelinating glial cell and the axon. However, when we looked more closely, we saw something that was remarkable. So I mentioned to you that potassium channels, these voltage-gated potassium channels, Kv1.2, are found beneath the myelin sheath in myelinated axons. And they're not normally found at the perinodal junction where the myelin sheath attaches. However, if you remove beta-2 spectrin, these potassium channels can now enter into perinodal regions, even though the perinodal junctions themselves remain completely intact. So what this means, what this experiment taught us, is that the paranodal cytoskeleton is the molecular basis of the paranodal barrier. Okay, so how do we so this suggests that there are two glia-dependent sodium channel clustering mechanisms. The first mechanism is a neurophasin 186 dependent mechanism that works through this extracellular matrix and glia-derived factors that actively cluster voltage-gated sodium channels. The second mechanism is a spectrin-dependent paranodal barrier mechanism. The paranodes themselves recruit a spectrin cytoskeleton that can function as a lateral diffusion barrier to limit the sodium channels between the adjacent myelin sheaths. Okay, so how do we prove that in fact these two glia-dependent sodium channel clustering mechanisms actually exist? I already showed you that if we get rid of one of them, sodium channels still cluster. If we get rid of this one, sodium channels still cluster. How do we prove that they both are functioning in tandem or together? Well, the way that we did this is we constructed a double conditional knockout lacking neurophasin and lacking beta-2 spectrin. So we had to construct mice that lacked the two mechanisms. And if we are correct, then we would predict that this should block the ability of sodium channels to be clustered at nodes of RON-VA. And indeed, that was exactly what we saw. So here you can see on top, these are the control. Sodium channels are clustered quite nicely, and paranodes are labeled here. But you can see there's a complete loss of voltage-gated sodium channels in these double conditional knockouts. There's loss of anchorin G and loss of beta-4 spectrin. And you can see the quantification here. There are about 30% of nodes that still have some sodium channel immunoreactivity. We don't know if that's due to poor recombination or not. But you can see here that there's a dramatic loss, arguing that these two overlapping mechanisms work together to assemble nodes of RON-VA. All right, so I've shown you that nodes and paranodes can have a mechanism that depends on beta-2 spectrin, and that nodes have mechanisms that depend on beta-4 and beta-1, as well as anchorance. But one thing that you'll notice is common between both of these, and that is the alpha spectrin. Alpha-2 spectrin is common between both the nodes and paranodes. So we were very interested to determine what would happen if we disrupted alpha-2 spectrin. And I already showed you one slide that if you completely remove alpha-2 spectrin, you get axon degeneration. But I didn't tell you what happens to nodes of RON-VA. So we constructed alpha-2 spectrin conditional knockout mice. And just like the beta-1, beta-4 double knockouts, these mice have severe ataxia. When we record sensory nerve conduction, they're dramatically impaired. Interestingly enough, and they are impaired on the rotarod as well. But interestingly enough, their neuropathic pain or nociception is completely intact. Nociception is not affected. It's only the myelinated axons that are, these are sensory, remember, so these are proprioceptors and mechanoreceptors. It's all of this phenotype is due to loss of mechanoreceptors and proprioceptors, which are the largely myelinated axons of the peripheral nerve. So what happens to nodes of RON-VA? So here's the wild type, and here's the conditional knockout, and you can see profound disruption. There's paranodes that will form with no nodes adjacent to them. When nodes do form, they usually only have a paranode on one side. It's dramatically impaired. Alpha-2 spectrin, it forms a periodic cytoskeleton at nodes, just like I showed you for axons in general. But this cytoskeleton is even more highly enriched at nodes of RON-VA. I think, and I don't have time to show you this, but one of the features that we learned about from these studies is that nodes of RON-VA themselves are particularly susceptible to injury, suggesting that the nodes are an Achilles heel for axon degeneration. Remember, we have this spectrin cytoskeleton that can function as a spring to maintain the flexibility of the axon. And as what you can see here is that nodes are a particularly important place to allow for that flexing and structural maintenance to occur. And in the absence of it, as I already showed you, you get axon degeneration, although unmyelinated axons remain completely intact. If we look more closely at the proprioceptors, so these are our proprioceptors that surround the muscle spindles. So these are these very interesting spiral windings around intrafusal muscle fibers. And what they do is they sense the tension and the stretch in muscle fibers to allow us to know and sense where our joints and limbs are located. So they're responsible for proprioception. But in these conditional knockout mice, you can see that all of these windings are completely disrupted and gone. So alpha-2 spectrin is absolutely essential to maintain axonal organization and structure, but principally of myelinated axons, not unmyelinated axons. So the last thing I want to tell you about is the function of glial anchorings and spectrans. So you've heard about anchorins at nodes of Ranvier in the axon. You've heard about beta-2 spectrin and how it participates in formation of a boundary, a barrier mechanism that can also help assemble nodes. I've told you about alpha-2 spectrin, which is required for node assembly as well as for axon integrity. But you'll see here in the myelinating glia, there's also beta-2 spectrin. It interacts with anchorins to assemble this important contact site. So to test that, we constructed glial beta-2 spectrin conditional knockout mice. And this was work performed by a former postdoc in the lab who now is running his own laboratory at Wright State Medical School. And here what you can see is we constructed beta-2 spectrin conditional knockout mice that lack beta-2 spectrin in axons. So the only thing you're seeing is the glial beta-2 spectrin. So this is all glial Schwann cell beta-2 spectrin, and it co-localizes precisely with anchorin B, as well as paranodal neurofascin 155. So what happens when we remove now this glial beta-2 spectrin? Well, if we look at these mice, I'm sorry if this appears a little small on your screen. Early on, so if we record nerve conduction velocity, what you can see is comparing wild types, hets, and conditional knockouts, is that these are significantly different between the wild type and the glial conditional knockout. In juvenile mice, it resolves in the adult, but then it gets much worse in the aged mice again. So this is very strange. It's bad in the young animals, it resolves, and then it gets bad again in the old animals. So how could this, what does this physiology reflect? Interestingly enough, myelination is completely normal. So myelin is completely intact. However, if we look at those paranodal junctions where the beta-2 spectrin is found, normally paranodal junctions are labeled by neurofascin 155, 186 is at the node, and it forms this beautiful structure. But at P13 in these glial beta-2 spectrin deficient mice, nodes are very disrupted. However, this kind of disruption resolves. And by 14 weeks, nodes look completely intact and normal, just like a wild type. So that explains this difference that we saw during early development, this kind of difference that resolves. How about this kind of difference? So if we look now in the aged animals, so comparing the adult situation with intact paranodes, now if we look in the aged mice, remember potassium channels are excluded from the sites of the paranodal junctions, but amazingly in these animals, the loops of myelin that are closest to the node of Ranvier itself, they detach and are no longer attached to the axon. So, and you can see that here, it's a little bit hard, but you see the transverse bands. This corresponds to the innermost axoglial junctions labeled here. But as you get closer to the node, the transverse bands that characterize an intact paranodal junction are completely gone. And that's what you see right here, these massive gaps where the myelin sheath has detached from the axon itself. And that's due to the absence of glial beta-2 spectrin. Remarkably, when Kai-Jun Chang, another grad student in the lab, generated glial conditional knockouts for Anchorance, he saw exactly the same phenotype. So these are paranodal, these are the cases with paranodes, shown here in green is the wild type, but it's massively delayed in the conditional knockouts lacking glial anchorance. But it eventually resolves, exactly like what we saw with the beta-2 spectrin conditional knockouts. So I think my time is about up and I want to be able to answer questions. So what I've told you today is that anchorins and spectrins can play diverse and key roles in many aspects of node of Ranvier biology. I've shown you that there are redundant and overlapping mechanisms where if you lose one of these molecules in the primary mechanism, that secondary mechanisms can compensate to still cluster voltage-gated sodium channels. I've shown you that there are two glia-dependent mechanisms, both of which, for assembly of nodes of Ranvier, both of which converge on the spectrin and anchorin cytoskeleton. And I've shown you that even in the myelinating glial cell, there are anchorin and spectrin-dependent cytoskeletons that are necessary to assemble and maintain nodes of Ranvier. So these are incredibly versatile and interesting giant proteins that evolved to help assemble the functional organization of axons so that we can have rapid and efficient axon potential propagation in our myelinated axons. With that, there's Mauricio when he looked very young. Now he looks a little older. But I want to acknowledge the people who have contributed to this work. Tammy Ho did all of the work using the Anchorin G conditional knockout. Kei Suzuki did the beta-2 spectrin work in glia. Kai-Jun Chang did all of the glial anchorin knockout work. Chuansheng Zhang did all of the beta-2 spectrin work. And Chengxin Liu did all of the work looking at the role of nodal beta spectrins. I'm sorry I don't have time to tell you about axon initial segments or this fascinating protein anchorin R that we found can compensate for loss of anchorin G, but that may be the stuff of another day. And if you can look on my board here, this is our paper on anchorin R, and I hope that soon you'll be able to read about it. So with that, I'll be happy to take any questions. And you can either ask questions of me directly, or you can also send them to the chat. So I have the chat here.
So I have a question, Matthew. Excellent talk. I really enjoyed it. It's regarding the anchoring R compensation. Is there a critical period for the compensation of anchoring G deficiency? And if so, is it, for example, reopened when there's damage?
That's a good question. So one thing that I did not tell you is that all of the things that I showed you today in the slides, even though we did them in peripheral sensory neurons, we did everything in parallel in the central nervous system. Now, the way that we did this in the central nervous system, we did it two ways. One was using a conditional Cre line, but the other way was using an adenovirus to introduce Cre. So to answer your question, what we did is we took adult mice, we injected into the vitreous of the eye an adenovirus to express Cre, and then we could visualize 2 months later nodes of Ranvier. They were GFP labeled, or the axons that were transduced were GFP labeled, and we could see that those axons had anchorin R present at their nodes of Ranvier to rescue the clustering.
Impressive.
So it can happen at any time. And the reason it can happen at any time is because anchorin R naturally exists in the axon. It's it's just not clustered because of the lower affinity.
Right.
So you may ask, what is the normal function of anchorin R in the nervous system? Well, that's
maybe substitute for anchoring G.
Why does it just substitute for anchorin G? That's right. It doesn't just exist to substitute for anchorin G.
Okay.
It has other functions.
I see. Thank you very much.
Thank you for the question.
Oh, thank you very much for the for the talk. A wonderful talk. The relationship between anchorin and spectrins is the same for any kind of sodium channels, for any kind of isoform of sodium channels. And the other question is, when you see these changes between young mice and old mice, is a difference between the isotype of the sodium channel that you find, because original are 1.2 and after start to be 1.6. So did you check for this, or there is any difference between these different kind of isoforms?
This is another very good question and interesting question. We do not see any difference in the isoforms. This is perhaps not surprising, because the mechanism for sodium channel interaction with anchorins is common between all voltage-gated sodium channels. There's a motif in the 2,3 linker domain of sodium channels. It's about a 21 amino acid sequence that is conserved across all voltage-gated sodium channels. And so all sodium channels can bind with high affinity to these anchorins. Okay, now, there are ways to modulate this. There's phosphorylation events that can happen that can change the affinities. So this is a very good question. In the experiments that we have done, we have not observed any subtype switching between the sodium channel isoforms. Now, I'll just maybe give you a teaser, and I will say that it turns out that there are differences. I didn't discuss any of this today, but there are also nodal potassium channels. And it turns out that even though sodium channels can bind to both of the anchorins, the kind of nodal potassium channel that is present is different depending on whether you have anchorin G or anchorin R. This is unpublished. If you have anchorin G, you have KV 7.2 and KV 7.3. These are the KCNQ2, Q3 potassium. You have KV 3.1b, another voltage-gated potassium channel. So it switches perfectly. Not only do you see a substitution in beta 4 for beta in the spectrin, but you also see substitution in the potassium channels that are present. So the kinds of potassium channels that are present at nodes are driven and dictated by the anchorins, not the kinds of sodium channels. Very good question.
Thank you very much.
Matt, have you checked what happened with the cytoskeleton in the different demyelinating model?
Ask that to me again. I didn't catch the first part.
Sorry, if you have checked what happened in the demyelinating model with the different cytoskeleton proteins in glia or in the axon.
So we have used a model. We've used lysolecithin as a model in the beta-2 spectrin glial conditional knockouts. Those mice fail to remyelinate as efficiently. They're far worse at myelinating. So even though developmental myelination appears not to be affected, remyelination is dramatically affected in the absence of glial beta-2 spectrin. But that's the only one that I can comment on.
Thank you. Beautiful talk. Jorge, there is more question?
I have a question, Matt. Very nice. Very nice talk.
Is this going to be a hard question?
I don't know. Another one. So you have the almost the full puzzle of the complex working on. I wonder, just thinking in the stability of this complex, how is the recycling of this protein, and have you measured, or do you know if there any research done on the stability of the complex comparing the muscular junctions and the nodes, for example?
Yeah, so actually this is those are two very good questions. So the first question is, do we know anything about the stability of the node and the recycling and reinsertion? So the person who's done most of the work on node recycling and insertion, this is work from Jim Salzer, and he just published a paper in Journal of Neuroscience a couple months ago, where he showed that the insertion and the recycling at nodes of Ranvier also depends on the paranodal junction. Okay, so it's also paranodal junction dependent. Now, your second question was, what about the neuromuscular junction? So I'm going to share my screen with you once more. Oh,
I will.
Jorge, can you
yes,
give me
yes, of course.
screen sharing again?
Here, go.
Okay, I'm going to share my screen again, because if I had had time, I was going to tell you more about the neuromuscular junction. All right, you can see this. So it turns out that sodium channels co-localize with Anchorin G at the neuromuscular junction. Okay, these are the only three places in the nervous system where sodium channels are clustered in very high density: axon initial segments, nodes of Ranvier, and the neuromuscular junction. And amazingly enough, what you can see is that Anchorin G is also present there. And so we proposed and hypothesized that the clustering of these sodium channels down in the junctional folds depends on a similar kind of protein complex that we find at nodes of Ranvier. Now, because of time, I'm going to skip through a whole bunch of slides. We had to generate triple conditional knockouts. We had to remove all three of the anchorins. If you get rid of one, sodium channels still cluster. If you get rid of just two, sodium channels still cluster. You have to get rid of all three of the anchorins in order to block the sodium channel clustering. So you can see here in the control, beautiful sodium channel clustering, but in these triple conditional knockouts, it completely blocks the clustering of voltage-gated sodium channels. So that probably answers your question, Mauricio. How is it that sodium channels are clustered at this site? We're just finishing this paper, and it's quite surprising that you can actually remove all of the sodium channels from the neuromuscular junction, and these mice, they don't care. They don't care. Okay? And the reason they don't care is because those sodium channels are not responsible for the main action potential of the muscle. What they do is they are there in order to respond to fatigue. So maybe just show you this. So if we stimulate repeatedly wild type and triple conditional knockout axons, so these are compound muscle action potentials stimulated at 60 hertz, you can see that these triple conditional knockouts that lack clustering of sodium channels only at the neuromuscular junction. They have sodium channels in the membrane, but they can't follow and they fatigue. Okay, that was a long answer to your question, Mauricio.
That's impressive. Thank you.
Matt, I have a question. So you show that the spectrins can compensate, but when they compensate, they use their own anchorings. So have you tried shifting? How is this affinity from, the anchorings on the spectrins?
We don't really understand the mechanisms of interaction or what the specific key interaction sites are that control why beta 1 spectrin likes anchorin R and why beta 4 spectrin likes anchorin G. We don't know that. We know the sort of general domains that are responsible for the interaction, but that's the extent of our knowledge. If you look at anchorin G, it can bind to beta 1 spectrin, but its affinity is much lower. If you look at anchorin R, it can bind beta 4 spectrin, but its affinity is much lower. So it seems that in some instances, The affinities themselves are what drive which proteins you find present at a particular site and structure.
Okay, but could they compensate in case the specific anchoring is lacking? So if you have the double lacking, of the spectrins, typical for this region.
Could we force them? We
Yes.
haven't done that experiment. So that's an interesting experiment. If we did an anchorin G conditional knockout and a beta 1 spectrin conditional knockout,
Yes.
now do we see anchorin R and beta 4?
Yes.
That's a very interesting question. That's a great question.
Great.
We haven't done that experiment. I like that very much. I'm going to take a note on that.
Okay. Thanks, Matt.
Any other questions?
No.
If not, then thank you for listening to me, and I envy you your spring.
Yeah.
Thank you, and thank you very much for accepting our invitation. And it was really nice at least see you through the screen.
And hopefully we'll all be able to travel again soon and visit with each other.
I hope so.
Thank you.
Bye-bye
very
and have
much.
a nice time.
Bye-bye. Thank you.
Thank you so much, Matt.